This presentation has been prepared for the Standards of Evidence for Life Detection Community Workshop. I'm Richard Quinn, and I'll be talking about the Viking biology experiments. In this presentation, I'll provide some background on the Viking biology experiments. I'll talk about the Life Detection Knowledge Base, which is a community tool designed to support discussion about the detection of signs of life. Talk about some of the Viking biology results and interpretations in the context of the KV structure, and then finally conclude with some thoughts for our upcoming workshop. To begin, I'd like to point out that during the time of the Viking mission, the way science and technology was developed for flight was in many ways the same as it's done today, including performing analog field studies and the development of instrument concepts. In the case of Viking, this included the biological capacity to generate metabolic carbon dioxide from labeled organics and the ability of soils to fixate labeled carbon dioxide. These instrument concepts were then further developed for flight as part of the Viking biology experiment payload under a demanding delivery schedule that included a preliminary design review in October of 1971, a critical design review in May of 73, and then in March of 75, the simultaneous delivery of the qualification unit, the biological payload for Viking 1, and the biological payload for the Viking 2 lander. Shown in parentheses is the number of months the delivery was behind the originally scheduled delivery date. The Viking Life-Detection Payload ultimately developed into 3 biology experiments designed to identify the presence of microbial metabolism in Martian surface samples. Each experiment used different induced test environments, which included dry to wet, light and dark, no additional nutrients, the addition of simple nutrients, or the addition of complex nutrients. For each experiment, a heat-sterilized sample served as the control. The Viking gas exchange experiment was based on the hypothesis that soil gas exchange kinetics could be used to determine the absence or presence of biology, and correspondingly was designed to measure changes in gas composition in the headspace above Martian samples. It was based on the assumption that gas compositional changes in soils are mainly the result of biology. Second, that metabolism in populations of soil organisms change when environmental conditions are perturbed. And finally, a complex nutrient medium could be used to alter the conditions and induce or inhibit metabolism. The design of the experiment was quite simple. A surface sample was placed in a test cell, the cell was sealed, and gas chromatography was used to monitor the headspace as a function of time under different conditions, which included dry incubation, a humid mode, and the addition of a complex aqueous nutrient medium. The Carbon-Assimilation experiment, also known as the Pyrolytic release experiment, is based on the hypothesis that Martian life would be carbonaceous and exchange carbon with the atmosphere. Correspondingly, was designed to measure the incorporation of radioactively labeled carbon monoxide and carbon dioxide into organic matter. The experiment could measure either photosynthetic or chemosynthetic fixation, and it approximated Martian surface conditions of pressure, atmospheric composition, solar radiation, and water abundance. In a pyrolytic release experiment, a surface sample was placed in a test cell, and after sealing, a small amount of radioactively labeled carbon dioxide and carbon monoxide was injected. After incubation, the sample was heated to 600 degrees C, driving fragments of any organics that had formed onto a sample trap. Excess labeled CO2 and CO was then purged from the system, and following that, the trap was heated to 700 degrees to decompose any labeled organics that had formed for detection as CO2 or CO. The labeled release experiment assumed that Martian organisms, if present, could assimilate carbon compounds and produce carbon dioxide or carbon monoxide as an end product. Correspondingly, was designed to measure radioactively labeled carbon dioxide or carbon monoxide evolved from the assimilation of radioactively labeled organics that were added to the soil. Metabolism and growth could be detected and rates could be determined by measuring radioactivity over time, and a positive result could be confirmed using a heat-sterilized control. Very simply, in a labeled release experiment, a surface sample was sealed in a test cell and a dilute aqueous mixture of simple labeled organics was added. Following that, the sample was allowed to incubate and the production of labeled carbon-containing gas was monitored as a function of time. With that brief introduction to the Viking biology experiments, I'd like to introduce the Life Detection Knowledge Base, which is an online community-owned resource to provide knowledge and support discourse about the detection of signs of life. These signs, known as biosignatures, consist of substances, structures, patterns, or processes that indicate the current or former presence of life. In the Life Detection Knowledge Base, information is presented according to arguments that support or contradict the value of a given biosignature as a sign of life, all organized according to criteria and supported by evidence drawn from the scientific literature. Currently, the astrobiology community is engaged in populating the knowledge base. If you'd like to become involved, you can find more information at the link shown at the bottom of this slide. When you enter the Life Detection Knowledge Base, you'll be asked to select a biosignature category. In the case of the Viking biology experiments, we'd select activity. The knowledge base is structured using criteria, where criteria are a set of standards against which to evaluate the utility of specific life detection measurements. Two of the criteria in the knowledge base are feature strength, which is the prominence of a feature that provides evidence regarding the past or extant presence of life, or a non-biological process, and feature prevalence, which expresses the likelihood that the feature is produced by a particular source, such as life or a non-biological process. And on the lower left-hand side of the slide, you can see that strength and prevalence criteria are categorized by biological feature strength, biological prevalence, as well as abiotic feature strength and abiotic prevalence, each along with corresponding pros and cons for a given argument. Now I'd like to briefly walk through the results of the Viking biology experiments and loosely place them in the knowledge base structure. There are precise ways in which arguments and their corresponding pros and cons should be crafted in the knowledge base, which I'm not doing here, nor am I including all the possible arguments and evidence related to the biology experiments that could be included in the knowledge base. Instead, I'm demonstrating the basic structure of the knowledge base that can be used to evaluate biosignatures. In the gas exchange experiment, when a sample was humidified or wetted, there was a rapid release of oxygen, which was disproportionately large relative to other gases released, including nitrogen and argon. A slow linear release of carbon dioxide was also observed, which persisted after a second injection of aqueous nutrients. Additionally, no significant changes were observed during prolonged incubation. This gas release pattern was observed for all samples tested, including stored samples, samples preheated to 145 C before testing, and a sample that was collected from under a rock. If we consider the hypothesis that the signal is biological, we can examine both pro and con arguments and the evidence that supports or refutes this assertion. Characteristics of the gas exchange response included that the reaction survives heating to 145 degrees C, the release of oxygen is extremely rapid, and a second injection of nutrient did not result in additional gas production, which indicates that biological growth did not occur. For the hypothesis that the signal is abiotic, we can again examine the results in the context of pros and cons. Post-Viking, it was shown that multiple plausible inorganic oxidants can explain the gas exchange experiment oxygen release. Also, post-Viking experiments showed that iron oxide can reproduce the carbon dioxide release. In the pyrolytic release experiment, all tested samples were statistically positive, which is indicative of the incorporation of radioactively labeled carbon dioxide or carbon monoxide into organic carbon. 9 samples were tested under different conditions that included dry and wet, light and dark, a sample that was heated to 175 degrees C prior to testing, a sample that was heated to 90 degrees C, and a sample that was stored for 139 sols before testing. Additionally, sample splits from the same sample collection, but tested under different conditions, shown in yellow, produced nearly identical results. If we consider whether the signal may be biological, the results show that heat treatment to 90 degrees C did not reduce the reaction, and heat treatment to 175 degrees C did not eliminate the reaction. If we consider whether the signal may be abiotic, the results show again that heat treatment at 90 degrees C did not reduce the reaction, and heat treatment at 175 degrees C did not eliminate the reaction. Additionally, post-Viking experiments demonstrated positive results using iron oxides, clays, and Mars analog mixtures, indicating that the Viking pyrolytic release results were false positives. In the labeled release experiment, when a dilute solution of simple organics was added to the sample, shown at 0 hours in the plot on the right, there was a rapid release of radioactively labeled gas, presumably carbon dioxide. A second slow log linear release of carbon dioxide was also observed. Significant gas release was not observed after the injection of additional organics. Additionally, the gas release was observed in a sample excavated from under a rock. The initial release was reduced by preheating a sample to 50 degrees C for 3 hours. It was eliminated by heating to 160 C for 3 hours. Also, the slow release was not eliminated by heating to 160 degrees. And finally, the initial release was observed from a sample stored for 3 sols, but not from a sample stored for 80 sols. For the hypothesis that the labeled release signal is biological, the results indicate that the initial release of gas resembled biological responses observed with the flight test module. Also, the initial release was not observed in samples preheated to 160 degrees prior to testing. On the other hand, as Chuck Klein has pointed out, the initial response would require a large biological load. A lag phase was not observed. The reaction lasts only about 24 hours. Only approximately 10% of the available carbon substrate is utilized, and injection of additional organics did not result in additional gas release. For the hypothesis that the signal is abiotic, the post-Viking literature shows that there are multiple plausible inorganic oxidants that can explain the labeled release results. The GEX results also provide evidence for the presence of oxidants at the Viking sites, which is also supported by the findings of the Phoenix and NSL missions. To conclude, I hope that this presentation can serve as a starting point for discussion at the upcoming workshop, in particular how the life detection knowledge base can be used as a tool to achieve both short-term and long-term workshop goals. Also, going back to the third slide in this presentation, a discussion on the risk associated with the application of standards to instrument verification and validation. This is a quote from Levin and Strott about the labeled release experiment. A science testing program was evolved to obtain data relevant to the interpretation of the flight experiment. However, because of program demands on the test standards module for engineering purposes, not as much science data has been collected as had been planned. I think that every scientist who has been involved with the delivery of a flight instrument has said these very words. And finally, a discussion on the risk of false positives. There was a tremendous amount of testing for biology and analog samples in preparation for Viking. But for the most part, consideration of abiotic feature strength and prevalence is absent in the pre-Viking literature. I'm looking forward to seeing everyone at the upcoming workshop and beginning a discussion on these aspects of developing standards for life detection. Thank you.